I completed my bachelor's degree in biomedical science with first class honours in 2013. Then between September 2013 and August 2014, I undertook a master's degree by research investigating the biochemical site of action of a group of anti-cancer drugs that were originally designed to stop blood vessel growth. I then went on to study for my PhD in the Department of Oncology and Metabolism at the University of Sheffield in September 2014, working under the supervision of Professor Ingan Holan and Dr. Penny Ottewell. My PhD project was focused on developing novel methods for studying the non-dividing breast cancer cells, which we refer to as quiescent or dormant tumour cells. My PhD was awarded in November 2017, and I've since been working as a postdoctoral research associate in the Holan Lab, continuing my work on understanding the biology of dormant tumour cells in breast cancer. The title of my current Against Breast Cancer funded project is Preventing Metastatic Disease by Targeting Breast Cancer Cells that Survive Chemotherapy. So in the UK, breast cancer is the most common cancer and claims nearly 12,000 lives each year. Around 2 out of every 10 patients will experience that their breast cancer spreads or metastasizes and returns several years after they underwent surgery, chemotherapy or radiotherapy. We know that this is because some cancer cells escape from the breast tumour and settle in other parts of the body, usually the bone marrow, where they can survive for many years becoming dormant or non-dividing and thereby remain undetected. In this dormant state, cancer cells aren't sensitive to common chemotherapy drugs, which usually target dividing cells, and at some point the dormant cells can be triggered to start growing again, and they can then cause spread to other organs such as the lungs, liver and brain. So in order to prevent breast cancer from returning, we need to understand how cancer cells become dormant and remain in this state so that we can develop new treatments that eliminate the dormant cells responsible for the cancer coming back. And this project will allow us to do that. So in this project, we'll use models that I've previously developed that allow us to specifically isolate the dormant breast cancer cells that survive treatment so that we can study them further. We're going to use advanced genetic study methods called transcriptomics, along with computer-based analysis, which is referred to as bioinformatics, in order to study the genetic machinery of the surviving cancer cells or dormant cells, and compare this to that of those that are sensitive to chemotherapy, i.e. the growing and dividing population. This will allow us to identify molecules that cancer cells switch on or off to become dormant and survive chemotherapy, and then we can check if these molecules are present in human breast tumours that we know went on to spread using information available in clinical databases, and this will allow us to select the most promising candidates for further study. So that initial study that I led on allowed us to establish that dormant cells in breast cancer, which I previously developed models to study during my PhD, are those that are able to survive chemotherapy and therefore begin to grow again after we stop chemotherapy. In this project, we aim to understand the biology that governs the generation and survival of those dormant cells. So that initial ABC-funded study really provided the final pieces of the jigsaw in terms of work that we'd already done, and now we can use those models in the really important work that this project will enable us to do in order to identify mechanisms that can be targeted to remove or aid the removal of the problematic dormant cancer cell population. The models that I use for studying dormant cells all rely to a greater or lesser extent on the retention of fluorescent dyes by cells that don't divide. So the fluorescent dyes that we use can be detected when we shine light of a very specific wavelength on the cells that contain dye, which makes them glow. We can then either use a microscope to see these glowing cells, or use a machine called a flow cytometer that can count the number of cells for us. So if you go on to imagine a population of cells where every cell is 100% full of fluorescent dye, each time those cells divide, they give rise to 2 new cells, each of which will contain half the amount of dye that the parent cell did. So after one division, every cell in a culture has 50% of dye. After another division, that halves again to 25%, so on and so on. So if we label a culture of cancer cells with fluorescent dye and allow them to grow continually, there comes a point at which the level of dye in the dividing cells will become so low that the fluorescence resulting when you shine light on them is no longer detectable. However, if the cells either don't divide after they receive their 100% of dye, or if they do so much more slowly than the majority of the cells in the rest of the culture, then they will still contain a large amount of dye, meaning that when we shine a light on them, which makes them glow, they will glow very brightly, allowing our machine to detect them. And if we use a slightly more advanced version of this machine called a fluorescence activated cell sorter, we can actually separate out the glowing cells, which are the non-dividing or dormant cells, from the growing cells. So most people will be familiar with the concept that cells contain DNA. DNA itself can be subdivided up into genes, which are basically the instructions for building all of the proteins that make the structures within a cell and for carrying out all of the functions that a cell does. In the case of humans, we have approximately 20,000 genes with known functions. So if DNA is the instruction and protein is the final functional product, the intermediate template that a cell makes is called messenger RNA. And messenger RNA does exactly what it sounds like, which is to take the message encoded in the DNA of a gene and form the template from which the cell can make a protein. Messenger RNA is only produced when a gene is needed or being actively used, and therefore more messenger RNA is produced when more of the protein encoded by a gene is required by the cell. So transcriptomic profiling is where we extract all of the messenger RNA from a set of cells, and following an experimental procedure called sequencing, we can determine which of their 20,000 genes a group of cells are using and to what extent. So in the context of this type of work, bioinformatics would refer to applying computer-based methods that we can use to analyse the data that we get from our sequencing experiments. So the bioinformatics that I undertake for these kind of experiments means that I write a series of computer programs which allow us to apply advanced statistical modelling methods to compare which genes are switched on or off in growing and dormant breast cancer cells, and map these into cell signalling pathways in order to understand the biological processes occurring in these two populations and how they differ. And we can also use bioinformatics to see whether genes or collections of genes that we identify as being different between these two populations are found to have the same pattern of expression in patients that we know went on to develop dormant metastatic disease. So collaborating with other experts is important as it allows us to draw from their knowledge base and the resources at their disposal, and this will ultimately vastly improve our capability and the work or the quality of results that we're producing at any one time. And this obviously works the same way when other people come to us. Open access data is also incredibly important for my work because when we've identified a gene or series of genes that we believe are important to cancer cell dormancy, we can go on to validate our findings in open access or publicly available data sets. For example, there are publicly available clinical data which are large collections of data where the follow-up information for a patient, so for example how long a patient survived or whether they developed secondary tumours, is available in addition to the data from sequencing of the tumours or tumours which they developed. So this allows us to look at a group of patients with high expression or low expression of a gene or series of genes that we've identified in our work as being interesting or important, and we can see whether this high or low expression is associated with a better or worse outcome. The results that we'll gather during this project will hopefully pave the way for future projects where we can establish whether stopping the dormancy associated genes from working using either genetic engineering or drugs reduces the capability of breast cancer cells to become dormant, thereby sensitizing them to chemotherapy. Ultimately, we hope that this work will allow us to develop new treatments to prevent breast cancer recurrence, benefiting a large number of patients who have disease for which there is currently no effective curative treatment.